Okay, so today is section 3 of our biochemistry unit, which is on carbon compounds. There are 4 main groups of carbon-based compounds that you're going to be responsible for knowing, along with their features and their characteristics and why they are important to biology. All of these groups of carbon compounds are part of a branch of chemistry that specializes only in the study of carbon-based compounds. These compounds contain primarily carbon and hydrogen. Those are the most used elements in these compounds. There are other elements that can be involved, especially schnapps, which we've talked about before, the other elements of life. But this branch of chemistry is called organic chemistry, and it centers around carbon. Carbon's really important because it can form large, complex, and diverse molecules just because of its structure and the number of valence electrons it has. It makes carbon a very versatile element, and it is the element that is the basis for all of life's compounds. So again, looking at carbon does have 4 valence electrons. So if we take a look and we draw a Lewis dot diagram of carbon, what we see is we start with carbon. It has 4 valence electrons, so there's going to be 1 electron on each side. And so that means that there are 4 bonding sites around carbon. This is important because that means carbon can form 4 bonds in all directions. That makes it so that it can form these chains that are very long, or also rings. Carbon can also form double or triple bonds between itself or other elements. So if you look up here on the picture, you see that there are some examples of double and even triple bonds between carbon. But the most important thing is that no more than 4 bonds can be around a single carbon at one time. And if you look at these double and triple bonded molecules, that holds true. So if we look at this carbon here, it's got 1, 2, 3, 4 bonds around it. If we look at this carbon, it also has 1, 2, 3, 4 bonds around this carbon. And then these carbons down here in the triple bonded molecule, if we look at this one, it's got 1, 2, 3, 4 bonds. And then that other carbon there is similar with 1, 2, 3, 4 bonds around it. So those 4 bonding sites really make carbon versatile for what we're going to see in these different carbon compounds that are very important for life. Because carbon can form these long chains, it can form these molecules that are very, 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 very big. We call those macromolecules. So if you look here at the title of this slide, you see the word macromolecules. Remember, if we break this down, we see macro and molecules. Well, we know what molecules are, but let's figure out what macro means. So macro is the opposite of micro. Micro means little or tiny, which means macro means big. Okay? These long chains and rings are formed by a process that is called polymerization. So these macromolecules are all considered polymers. Okay, remember the prefix poly means many. So all of these molecules are formed by joining individual building blocks together to make a big molecule. So we can break these molecules down into their tiny little individual bricks, which are called monomers. Mono, remember, means 1. The prefix mono means 1. So when we join individual monomers together to make a polymer, we call that process polymerization. So in forming these polymers with different building blocks, we make four main classes of biological macromolecules, or big molecules that are responsible for life. These four groups are carbohydrates, proteins, lipids, and nucleic acids. And we're going to go through each of these one by one and tell you some characteristics and the function of each of these molecules in your body and in all living things. So the first group of molecules we're going to look at are carbohydrates. Carbohydrates are important in your body because they are the fuel and the building material, and also they provide short-term energy. Okay, short-term energy is energy that's used over the course of a day, maybe a couple days, in your cells and in your body. If you're an athlete, you're really familiar with this because a lot of sports, especially here in the school, have pasta dinners the night before big games. Football team does it, track team does it, swim team does it. A whole bunch of sports do it organized, and if your sport doesn't do it in an organized fashion, a lot of the coaches you'll probably hear say, go home and eat a good dinner with carbohydrates, because carbohydrates give you a good amount of energy that you can use the following day in your sporting event. Carbohydrates include things like simple sugars, which are things like fructose, galactose, glucose. Okay, all of those are monomers or building blocks of big carbohydrates. One of the biggest carbohydrates that is a storage carbohydrate is starch. Okay, starch is stored in all of those carbs that you eat on a daily basis, and it is the big version. So we take these Lego bricks, these monosaccharides. Okay, so the monosaccharide is the monomer of a carbohydrate. So it is essentially the Lego brick that we're going to build these complex carbohydrates with. So one of those is called a monosaccharide. When we hook lots of them together, that's called a polysaccharide. So again, if you look at the prefixes in the words, mono means one, poly means many. Carbohydrates also have a fixed ratio. That ratio is 1 carbon for every 2 hydrogens for every 1 oxygen. So it uses things in that 1:2 ratio. And if we look at that, it makes sense. So 1 carbon for every 2 hydrogens for every 1 oxygen. Well, let's break this apart. We know H2O is water. And we know that just C is carbon. Well, what's another word that you use when you drink water? You're hydrating yourself. So if we look at the name of this molecule, it's a carbohydrate because literally it is a hydrated carbon because of this ratio. So we hydrate carbon to get a carbohydrate. Now let's look at the process of hooking these monosaccharides together. So I've told you that monosaccharides are like the Lego brick of a carbohydrate or a polysaccharide. We take these little monosaccharides and we hook them together, one after another, after another, after another, to make these big molecules. Well, we can also just hook 2 together, and when we hook 2 together, the name for that molecule is a disaccharide. Okay? An example of a disaccharide is sucrose, that you've probably heard of. So when we hook these monosaccharides together, we use a process that is called dehydration synthesis. That's kind of a big name, but if you break it down, it's not too hard to remember. Dehydration means that we're removing water. If you're dehydrated, you are lacking water. So in dehydration synthesis, we remove water to make something. And a fancy sciencey word for making something is synthesis. Okay? To synthesize something means to make something. So dehydration synthesis literally means to make by removing water. And if we look down here at this figure, we see an example of dehydration synthesis. So here we see glucose and fructose. Okay? And when we look at these molecules side by side, you'll see here that these parts of the molecule, the glucose has this OH group here on the one side of it, and the fructose has this hydrogen here on its OH group. Okay, now what we're going to do is we're going to remove that OH and H. And remember, if we add hydrogen to hydroxide, which is OH, we get water. So if we remove that water, we open up this oxygen here to be able to form another bond. So if we remove the hydrogen, this oxygen has a bonding site that's free. So this oxygen is basically going to reach his hand or his arm out, his bond out, to bond with this other molecule here. And you can see that in the resulting sucrose molecule. Okay, so when we remove the water, we get this bond formed here, and that special bond is this bond right here that is formed, hooking those two molecules together. In the process, we've removed water from the situation. And again, that is dehydration synthesis. Now, can we go the other way? Can we take this disaccharide and break it apart into two monosaccharides? And the answer is yes. We do this by adding the water back in, okay? That is called hydrolysis, okay? Now, that word kind of breaks apart too. If we look at it, obviously you see the word hydro there, meaning water, okay? And in science, the word lysis means to burst. Okay, if we say that a cell has undergone lysis, we mean the cell has literally burst. So when you add that together, it's the process of bursting with water. So you're not really bursting the molecule, you're just breaking it apart into these two little pieces, these two Lego bricks, the monosaccharides. But that is what the other direction is called. We add the water back in, we put this water back in here, we add the water back into the molecule, and it breaks it apart into these two molecules over here. Now, if we take those monosaccharides and we join lots of them together over and over and over and over and over again through dehydration synthesis, this gives us what is called a polysaccharide. Okay, and a polysaccharide is many monosaccharides together. It's a big carbohydrate molecule. If you look at the word, saccharide is a fancy word that means sugar, and poly means many, so it's many sugars put together. Okay, and starch here is one of the main polysaccharides. It's one of the most common. And you can see here, each of these little kind of circle-looking things, that's a monosaccharide. So you can see just how many of those monosaccharides are all hooked together there to make that starch molecule. So again, when we look at how these molecules are made and broken down, we make them by dehydration synthesis. We break them down by hydrolysis, okay? Hydro, again, means water. Lysis means disintegration, breaking down, bursting, all of those things. So when we put that word together, it means that we are breaking something down by adding water. This is how your body breaks down the carbs into the monomers to use as building blocks for other complex molecules. So think of it kind of like this. When you take in food from other sources, your body has to take those big complex molecules in, say, the bread that you ate, and it has to break that molecule down into these individual monomers, these Lego bricks, so that your body can then take those Lego bricks and build them into a molecule that your body needs. So you as a human don't store your energy in the form of starch. You actually store your energy in the form of glycogen, which is basically the animal kingdom's version of starch. So it's a big complex storage molecule. But when you eat fruits and vegetables and other plants, you're taking in the starch molecule. So your body takes that starch molecule, breaks it down into individual Lego bricks so that it can use those building blocks, those Lego brick monomers, those monosaccharides, to build up a complex glycogen molecule. And that is the basis of metabolism. And on this graphic, you can see hydrolysis taking place. Up here, we've got the disaccharide of sucrose. And what they're doing here is they're adding water to the sucrose into this chemical reaction. And that water breaks apart this bond here. And they join back with their old partners. And this hydroxide bonds to the glucose, and the hydrogen bonds back to its oxygen buddy, and you get the separate monomers or monosaccharides of glucose and fructose. So what are some of the most popular examples of different polysaccharides in life? Well, I've already talked about starch. Starch is how plant cells store most of their energy. And I've also talked about glycogen. Glycogen is the animal version of starch. So it's sometimes called animal starch. It's how energy is stored in your animal cells. You're a member of the animal kingdom. Your body is made up of animal cells, and you store your energy in the form of glycogen. Starch and glycogen are a class of carbohydrates that are called storage carbohydrates, meaning that they store things, they store energy. There are also a class of carbohydrates that are called structural carbohydrates. The two big examples of those that I'm going to give you, one of which is cellulose. Cellulose is found in the cell wall of a plant cell. So if you remember back to middle school, I know it's a long time ago, but if you remember back, you learned two types of cells, animal cells and plant cells. And we'll re-look at that when we get to the cell unit. But one of the main differences is that plant cells have these rigid cell wall. And so the cell walls are like these rigid squares that keep the shape of the plant cell. These cell walls are primarily made up of cellulose. That's what helps give it its rigid structure. So it is a structural carbohydrate. Another structural carbohydrate is called chitin. Chitin forms the exoskeleton of insects. So you find the cicada shells in the summertime, and you step on a bug and you hear that kind of crunch. That's because bugs have this exoskeleton on the outside, and one of the main structural carbohydrates involved in that exoskeleton is chitin. Okay, so we finished up carbohydrates, and now we're moving on to the next class of biological macromolecules, and those are lipids. Now, within lipids, there are four other main classes of lipids. The first being a triglyceride, which is a fancy word for fats. Fats are responsible for storing long-term energy. Okay? That should make sense because, when a bear is getting ready to hibernate for the winter, one of the things it'll do in preparation for that is it'll put on a significant amount of weight. That weight and that fat is there to provide that bear with energy throughout the winter. in case food gets scarce so that the bear can still survive. Fat is also there for insulation, helps keep things warm. That's why animals that live in cold places on Earth, in the Arctic and such, have an excessive amount of fat. It helps insulate their body and keep them warm. And the next class of lipids are steroids. Steroids are chemicals that regulate things in your body. Cholesterol is an example, and there are some hormones in your body that are also steroids as well. Another class of lipids are called phospholipids. These are really important in the cell membrane, and we'll talk about them a little bit now, but we will talk about them a lot when we get into talking about the cell membrane. The cell membrane is very important in lots of functions of the cells. And the last class of lipids are waxes. Waxes, primarily their function is to protect from water. So when you touch a leaf, you'll feel that waxy coating that's on the surface of the leaf. That is there to protect the leaf from water so that the leaf doesn't get waterlogged. That's one of the reasons why when you look at those pretty pictures of the morning dew resting on the leaves, you get these little balls of water. Okay, it's just like in our properties of water lab, you put the drops of water on the wax paper and you saw how the water was kind of afraid of the wax and it balled up in that tight little ball because of cohesion. The same thing happens on the surface of a leaf. The wax is there to protect the leaf from being saturated with that water, and so the water will just ball up in a delicate little droplet on the leaf, and it makes for a very pretty picture, especially as the sun rises and hits off of that dew on the leaves. But primarily it protects the leaf from becoming too waterlogged. Some facts about lipids: they are made primarily of carbon and hydrogen. They are nonpolar. That means they are very insoluble in water. Remember, insoluble means they do not like water. They don't dissolve in water. They, in fact, are very hydrophobic. And that's a fancy science word that means they don't like water. And if you break that word down, phobic makes you think of a phobia or a fear. Hydro, again, water. So when you break that word down, it means like water fearing. And nonpolar molecules they tend to be hydrophobic. They don't like water very much. And lipids also account for about 40% of the organic matter in the average human body. So we're going to look at the structure of fats here for a minute. The monomer of a fat is kind of two pieces that are coming together. You get fatty acids and glycerol molecules. So here is a glycerol molecule. It is this vertical chain here. And you get these 3 fatty acids here. So those are the building blocks that make up a lipid. We're going to form this lipid in the same way that we formed a polysaccharide. We're going to use dehydration synthesis. Okay? So if you look down here at this graphic, you'll see we've got OH here, here, and here. And then we also have another H over here. So we're going to join that together and remove the H2O. We're going to join that together and remove the H2O. And we're going to join that together and remove the H2O. That's going to result in 3 removed waters, and it's going to result in 1, 2, 3 new bonds that are formed in this fat molecule. Okay, so you see these bonds here that have been formed due to the removal of these 3 water molecules. 1, 2, 3. So if you look back at the slide that we just finished, when we built that fat molecule, we had that vertical glycerol molecule. And then coming off of that molecule, once we created the fat, we had these 3 fatty acid tails that kind of came off of that molecule. Those tails contain carbon bonded to hydrogen primarily. And when those tails contain the maximum number of hydrogen atoms that they can, and only single carbon bonds, they're called saturated fats, meaning that that fatty tail or that fat has the maximum number of hydrogens. Remember, carbon can form up to 4 bonds. So if we're forming a chain of carbons, 2 of the bonds here are taken up by bonding to other carbon atoms. But that means we have 2 bonding sites left. So we can use those to bond singly to 2 hydrogen atoms. And that's typically what happens in organic chemistry. You see these carbon-carbon bonds, and then hydrogens kind of fill in all the other bonding sites on carbon. However, sometimes you can get a situation where instead of this, you would have that carbon, but it would be double bonded to the next carbon. If that's the case, if you look here, we've used 1, 2, 3 of carbon's bonding sites already. So that means there's only 1 more bonding site where we can put a hydrogen. So this molecule over here does not have the maximum amount of hydrogens that it could because of this double bond right here. This molecule over here, however, does have the maximum number of hydrogens around this central carbon because there's only single bonds to the other carbons, so the maximum number of hydrogens I could fit in there would be 2. In this other example, this central carbon has used up an extra bonding site by double bonding to this other carbon over here. In using that up, we can only fit 1 hydrogen around that carbon, so that would be considered an unsaturated fat because it has at least 1 carbon double bond. If the fatty acid tail has more than 1 carbon double bond, it is called a polyunsaturated fat. Okay, remember, poly meaning many, so there's many unsaturated carbons in that chain, and so we call it a polyunsaturated fat. Now, structurally, being able to tell the difference between a saturated fatty acid or fat and an unsaturated fatty acid or fat is pretty easy because once you have that double bond between those carbons, it causes a kink in that fatty acid tail. So if you look over here to the right, we've got this diagram that shows a saturated fat. Notice how the 3 tails here, 1, 2, 3, are all perfectly straight. That's because if you notice up here, there are only single bonds between these carbons. Single bonds, or 1 bonding stick when you draw it out. Once we add in a double bond, you'll see that these fatty acid tails get these bends in them. So you see here how these ones have these bends going down the tail. That is an unsaturated fat. Now, unsaturated fats are actually more healthy for you than saturated fats. Saturated fats are like butter and things that are more solid fats, whereas unsaturated fats are more of the liquidy olive oils, those kinds of fats. Now, that being said, everything needs to be taken in moderation, but that's why they always tell you you should cook with olive oil instead of cooking with butter or lard, because the olive oil is a little healthier for you. So the next group of lipids we're going to look at are phospholipids. Phospholipids kind of look funny. They are primarily responsible for membranes within your cells. So if you look here, there's a piece of a membrane that's kind of been drawn out over here. So when we look at that, if you can envision this, this membrane would continue around the entire surface of the cell. So this dual layer is going to kind of continue around to envelope in the cell in a complete three-dimensional, almost ball of these phospholipids. Now, phospholipids are made up of a structure like such. They start out, they have a head. Oops. Let's redraw that. They have a head, and then they have these tails that come off of them, and they line up next to each other like such. Now, these heads and tails are two different parts with two different sets of characteristics. The heads are actually what we call hydrophilic. Okay, hydrophilic means water loving. Okay, the tails here are actually hydrophobic. That we already kind of talked about that word a little bit. Hydrophobic means water fearing. So here you've got those two vocabulary words and their definitions. These phospholipids make up the membranes of cells and organelles. And if you have trouble remembering which is which part is hydrophobic and hydrophilic, you can just kind of imagine that if this here is the membrane of a cell, the cytoplasm is going to be right up against those heads of the phospholipids. Cytoplasm, again, is that jelly substance that is inside of cells that, again, you should have learned back in middle school. That watery cytoplasm is going to be right up against the heads of these phospholipids. And if the heads didn't like water, that would be really bad for them. But lucky for the cell, the heads of the phospholipid are water loving. So they're going to line up next to that cytoplasm. Whereas the tails here, they don't like water at all. So these phospholipids are going to orient themselves to where the tails flip around and create this kind of hydrophobic zone in the middle of the membrane. These guys don't like the water in the cytoplasm, so they're going to line up to get as far away as possible from that watery cytoplasm. Okay, on this slide you can see the basic structures of some common steroids within your body. Steroids are formed from 4 fused rings of carbon atoms. Now, before you get all freaked out about what these structures look like and trying to wrap your head around what they mean, I can tell you this. This is how they draw molecules in organic chemistry. But what's happening here is because they don't want to draw out all the carbon atoms and all the hydrogens, it gets very exhausting to draw out all of them. Anytime 2 lines meet in a point, there's a carbon atom there. So if we look at this structure here, this cholesterol, there are carbon atoms at each of these points along that hexagon. And if you don't see something else drawn on the carbon, so here we've got carbon bonded to this OH group here. Remember, that's 1 bond for that carbon. Then we come over here, there's another bond here, that's 2 to this next carbon. This is 3. There's only 3 bonds around that carbon. So that means that there's another bonding site where we can just assume that a hydrogen has kind of jumped in there. Okay, hydrogen will jump in and fill in all those empty bonding sites for carbon. So when you look at this structure, again, anytime lines meet or form a point, there's carbons there. Here you can see there's a double bond in this structure, and you can kind of mentally fill in all of these carbons along here. And then anytime you see a carbon, count how many bonds are around it, and however many bonds you don't see, that's how many hydrogens are surrounding that carbon as well. So here we have this carbon that only has this one bond here. So that means out here there are 3 other bonds to hydrogens to get that carbon to its 4 bonds. Okay, so that's kind of how you read those structures. Again, it would take forever to draw this out and fill in all of those hydrogens. So whenever you see a structure like this, you can just assume that any place on the carbon that doesn't have a bond with its 4 bonds, there are hydrogens filling in those gaps. So these are steroids, and they kind of look like chicken wire if you're aware of what chicken wire looks like. But it's important that you kind of recognize these structures. You don't have to memorize the structures. I'm not going to ask you on the test, draw me cholesterol. But it is feasible that I might have a fatty acid and a phospholipid and a steroid, and I could say, you know, which one of these molecules is a steroid, and you need to be able to recognize that it's the one with the 4 fused rings of carbon. Okay, and so you can see them here, 1, 2, 3, 4 carbon-based rings that are fused together. These examples that I am giving you here are estradiol, testosterone, and cholesterol. Those are 3 main examples of steroids in your body. Next, we're going to move to our next macromolecule class, which are proteins. Proteins are extremely important in your body. They perform lots of functions, and they account for 50% of the organic material in your body. They are involved in so much. It's crazy. We're going to learn later on this year how proteins are made. Proteins are actually encoded in your DNA. So you know that DNA is kind of what makes you, your genetic code. It codes for everything that happens in your body. And that DNA code is the instructions for building proteins. So where DNA kind of gives you the blueprint, the proteins are what are actually doing the jobs in your body. They are actually performing the tasks encoded in the blueprint of your DNA and making you. Proteins include all of the elements in schnapps except P. So that can kind of be used to your advantage to remember. So if you remember schnapps and remember that protein starts with P, and now take P out of schnapps, those are the elements that you're left with that make up proteins. So carbon, hydrogen, oxygen, nitrogen, and sulfur. So what's the building block of an amino acid? Just like lipids and just like carbohydrates, proteins are made up of lots of these monomers hooked together. These monomers for proteins are called amino acids. Okay, and those amino acids are hooked together in long chains to form proteins. There are actually more than 20 essential amino acids that have been discovered. Some of these amino acids can be built by your body. The others cannot be built by your body, and they have to be taken in your body through your diet. So that's why protein is an essential part of a balanced diet, because you have to take in that protein so that you get those other amino acids that your body can't build on its own. Okay, and those amino acids are hooked together to make the proteins of life. Again, it's just like the carbohydrates, in which case you eat the protein. So you eat a chicken breast for dinner. That chicken breast contains proteins that were essential for the chicken, not necessarily proteins that are essential for you. So your metabolism, your body is responsible for taking those complex big chicken proteins, breaking them down into their amino acid building blocks or Lego bricks, and then your body is going to take those bricks and make a protein that you need, something that is essential in your body. And that is what your metabolism does. It breaks down the food that you eat into those building blocks and uses those building blocks to hook them together to make something new and a protein that you need. All amino acids have a similar structure. They start out with a carbon in the center. We call that the alpha carbon. If you think about like a dog pack, the alpha is the leader. It's the center of everything. The same thing is true with an amino acid. The alpha carbon is that carbon in the center. And then you have groups around the outside. You've got a carboxyl group on one side, an amino group on the other side, and an R group that changes based on what amino acid you're talking about. So the rest of the structure of the amino acid stays exactly the same. And the amino group and the carboxyl group are the groups responsible for hooking together to make a protein. Okay? So here we actually see a picture of this structure in action. So you've got your alpha carbon in the middle. Okay, that's this guy. And then you've got your carboxyl group on one side and your amino group on the other. And then pointing down below, you've got that R group. That R group is the variable group. It's going to change based on what amino acid it is that you're talking about. It is important that you know these groups around the amino acid. So if you look here, there are some examples of the amino acids. So these are the amino acids that make up the proteins in your body. And you'll see that each of these amino acids has the alpha carbon in the middle, the carboxyl group on the one side, the amino group on the other, and then you see the blue box, at least in this top group, it's blue. Down here, that is the R group. That's the variable group. You'll notice that in each of these amino acids, glycine, alanine, valine, leucine, isoleucine, if you look at this R group here, it changes in every single amino acid that you're looking at. But the thing that stays the same, the carboxyl group, the amino group, that central carbon, and then just like it always does, there's a hydrogen that fills in the gap. Because remember, carbon has to have 4 bonds around it at all times. Now, proteins are a little weird in that they've got a more complicated structure than some of the other macromolecules. So when we hook amino acids together, we're going to do that by dehydration synthesis. So we're going to take the OH group from the carboxyl side and the hydrogen from the amino side, and we're going to remove that to make water in our dehydration synthesis process, and that's going to form a bond here between the amino group of one amino acid and the carboxyl group of another amino acid. That bond is called a peptide bond. And as we hook these amino acids together, one after another, after another, after another, we make a long chain of these amino acids. That is called a polypeptide. Okay? Now you might be thinking, well, why isn't that just called a protein? And here's why. It's not called a protein because the long chain of amino acids then has to fold together into this kind of ball. Once those amino acid chains have folded into a ball in a specific shape, that is called a protein. But until it does that, and if it's just a long chain of amino acids, we call that a polypeptide. So again, we take these long chains of amino acids, these polypeptides, and we fold them into precise 3D structures that carry out functions. You can think of these like 3D puzzle pieces. In your body, your proteins have to have a specific shape so that they can fit together to carry out these functions. If the protein has the wrong folded ball shape, it's not going to do the proper job that it needs to do. So this structure of proteins is very important. So when we're building a protein, and we're going to look at that even more when we look at protein synthesis later in the year, but your DNA codes for this long chain of amino acids called a polypeptide, and then that polypeptide is going to fold into the protein that's going to carry out a job in your body. So here you can see the steps in making a protein. You've got the primary structure, the secondary structure, the tertiary structure, which is the third step, and the quaternary structure, which is the fourth step. So in the primary structure, we've just got, like you see, a long chain of amino acids. Okay, that long chain of amino acids hooked together. Each of these little circles is an amino acid. We hook all of those amino acids together by dehydration synthesis, and we've made a polypeptide here. That is the assembly process. Then we move to the secondary process where it begins to fold. So you get these foldings of those amino acids, and they curl around and they fold next to each other again, and they keep condensing into the folding process, and then they take their folded selves and they kind of pack it all together into a specific ball. That packing is done in the tertiary step of protein structure. And then we get more than one of these packed proteins that come together. So here you can see one packed protein is interacting with another made and packed protein. This interaction is the quaternary structure, and that's when the protein begins to do its actual job. Now, you've heard me tell you that proteins are super important in your body, and they do all kinds of different things. But you're probably like, what are some examples of these things? So here are the 6 main uses of proteins in your body. And I'm going to give you a kind of a funny saying on how to remember these 6 uses, a mnemonic device, if you will. But first, we're going to start with explaining these different uses. The first use is structure. Okay, collagen, keratin, these are all proteins that are involved in your skin and your muscle and building up that muscle. You know that when you lift weights, some people drink protein shakes after they work out or after they lift weights. The protein is involved in building up the muscle. That's why people do that. So we see that proteins are involved heavily in the structure of your body. Proteins are also used for transport. Okay? Things are transported through your body using proteins. Okay? Things are brought in and out of the cell through these protein tunnels. But also, you've got this really important protein in your bloodstream. It's called hemoglobin. And that protein is designed to bind to a molecule that I think you'll find kind of important. That hemoglobin binds this little molecule right here. That molecule is oxygen. It's very important for you because you need oxygen in order to carry out functions within your body and for your metabolism to kind of take place to create energy for your cells. Oxygen is very important, and it's moved through the bloodstream by a protein called hemoglobin. The next use of protein is for defense. The antibodies in your blood that are circulating throughout your body are proteins. These antibodies help you remember that last cold that you just got over so that when you encounter that virus again, your body can fight it off easily. Antibodies are like acquired roadmaps on how to defeat these pathogens or these bad guys that try to make you sick and invade your body. The next thing that proteins are responsible for is movement, muscle movement, cellular movement. Okay, so on a cellular scale, your cells have to be able to move in your body. That is done by protein filaments. The 2 main ones are actin and myosin that are involved in the filaments of your body, in your cells. And then also, you've got your muscle movement and how you move throughout your day-to-day life. Proteins are involved in that process as well. The next one is a little weird, catalyze. If you don't remember what a catalyst is a molecule that speeds up a chemical reaction. Okay? And these catalysts in your body that are proteins are called enzymes. And we're going to talk about enzymes in the next section of this unit in depth. But enzymes are super important because your body wants to be the most efficient that it can possibly be. And enzymes allow these processes in your body to happen without a large amount of energy input. So enzymes are proteins that are involved in pretty much every chemical reaction that takes place inside your body. And lastly, regulation. Your body regulates itself throughout the movements of chemicals like steroids or hormones, and these regulating chemicals do things in your body. Now, we know that steroids are lipids, and yeah, they're involved in the regulation process, but there's also another group of hormones in your body that regulate your body functions that are proteins, okay? So they're extraordinarily important in the day-to-day functioning of your body. So if we look at all of these together, we get these letters. So if we use the first letter of every function, we get S, T, D, M, C, R. Okay, now I've made up kind of a funny saying to go along with this. If you've been in health class, you should know this to be true, but STDs may cause rashes, which we know to be true from our health classes here in high school. But STD, structured transport defense, may, movement, cause, catalyze, rashes, regulate. So you can kind of remember that saying to help you remember these 6 uses of proteins. Proteins are very temperamental. They only work in very, very specific situations, and that is where our pH comes back into play. So proteins can only do the jobs that they do if they are within their optimal or their best range of temperature and pH. This is why your body needs to stay at a certain temperature all the time, because the proteins in your body, the enzymes and the other proteins that do all those jobs that we just talked about, have to function at the temperature that your body is at and the pH of the different parts of your body. So for instance, the proteins that are involved in the breaking down of food in your stomach only work at that very, very low pH because of the stomach acid. So if you took the proteins out of your stomach and put them somewhere else in your body where the pH wasn't that low, they're not going to function right. Same thing with temperature. When your body heats up or cools down because you're in a hot environment or a too cold of an environment, that's why you struggle to survive in environments that are too hot or too cold for an extended period of time without being able to warm yourself up to keep yourself at that constant body temperature. And that's why it's dangerous to be out, say, in the cold for a very long period of time, because it can affect the functions of your body, and it can eventually lead in death if you are, say, out in the cold for stranded for days without a coat and without any way of making a fire to heat yourself up. Hypothermia is going to set in, and you're going to get cold, and your body is just going to start to shut down because it can't function at those low temperatures. Similarly, if you have very, very high fever and your internal temperature is going up and up and up and up and up, it's going to get too hot inside your body, and your proteins aren't going to function at that temperature. And so that's why, it's very important. And doctors really focus on, once the fever gets to a certain point, they really focus on trying to get that fever down and bring the body temperature down. Because if the temperature and pH are not in their optimal range, the protein can do what is called denaturing. Okay, and denature just means a fancy word for unfolding. So that complex 3D structure we talked about will just break apart and fall apart. And if it's not in that structure, that complex ball that it's been folded into, it's not going to function properly. Okay, so denaturing is a result of being outside of that pH or temperature range that is optimal for those proteins. Okay, our last group of macromolecules is the nucleic acids. Nucleic acids are macromolecules that contain all of CHOPS except for sulfur. They don't contain S. So SNOP, singular. Carbon, hydrogen, nitrogen, oxygen, and phosphorus are involved in nucleic acids. The monomer of a nucleic acid is a nucleotide. There are two types of nucleic acids in living things. There are DNA molecules, which stands for deoxyribonucleic acid, and there are RNA molecules, which stands for ribonucleic acid. Those two molecules, the function of them is to store and transmit all of your genetic and hereditary information. So your DNA contains the code for everything that makes you you, and everything that has been passed down from your parents to you to make you is encoded in your DNA. And that DNA holds the code, and then it uses RNA to help make the proteins. So these two molecules are the only nucleic acids in your body. The building block of the nucleic acid is the nucleotide. So when you're looking here, this is the structure of a nucleotide. Okay, it's made up of 3 parts. It has a phosphate group, a 5-carbon sugar, and a nitrogenous base. Okay, and we'll get into depth with that a lot more once we get into our DNA and RNA unit. We're going to study the structure of this thing really in depth. But these are the 3 parts of a nucleotide. You should be able to recognize the nucleotide, and you shouldn't be able to recognize the 3 parts of it. Pretty easy. The 5-carbon sugar looks just like what those monosaccharides look like in our carbohydrates, but it's hooked to a group that has a phosphorus in the middle. That phosphorus is the phosphate group. And then it's also hooked to this single or double ringed structure up here that you see has a lot of element N in it. Element N, of course, being nitrogen. So that is the nitrogenous base part of the nucleotide. And we're going to hook those nucleotides together to build the molecules of DNA and RNA. And here you can see those 2 molecules of DNA and RNA. DNA, of course, has the double-sided, double helix structure. It's very famous. I'm sure you've seen it before. And RNA is actually a single-stranded structure. It's exactly the same as DNA, except for that you'll see it trades out the base thymine here for the base uracil. And we'll look at that more in depth later on in the year. But this single-stranded structure of RNA is like it's just half of the DNA molecule. So DNA actually has 2 sides to it and is double-stranded, but RNA is only single-stranded. So for right now, what I need you to know is that the examples of nucleic acids are DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. The building block of these nucleic acids is called a nucleotide. And that nucleotide is made up of those 3 groups, the phosphate group, the 5-carbon sugar, and the nitrogenous base. And that the function of these molecules is to store and transmit or send out instructions for your genetic and your hereditary information. Okay, so we have gone through the 4 main groups of biological molecules. We're going to do some activities with these in class. If you have any questions, please feel free to email me. Stop in and see me in my classroom. Come up after class, during house, whenever. I'm happy to help at any time. But this has been section 3 of biochemistry on carbon compounds. And our next section will be on enzymes. And that will be our last section of the unit. Okay, so until then, keep studying your biology, and I will see you in class.